this part is going to be showing you how we process the data, how we integrate the data to make them comparable between genes and between species. Okay, so, and I'm going to be using first the example of single-cell RNA-seq data. So as a reminder, I just present briefly how do we obtain single-cell RNA-seq data so that then you can see how we process and integrate them. So as a reminder, so to generate droplet-based single-cell data, so you have beads to which are attached like a sequence with the primer for the PCR, a cell barcode that will allow to identify each cell from which we have sequencing read, a UMI, a unique molecular identifier to identify each transcript, and a poly T tail to attach to the poly A of mRNA. Then it goes through a specific device, and each cell is going to be trapped into a droplet. And ideally, you're gonna have one cell with one bead in the droplet, so that then you can do the sequencing part. Uh, so you're gonna have like each cell isolated to obtain the information about the gene expression in each of these cells. And then you're gonna have a bioinformatic pipeline so that you can distinguish these cells in different clusters. And then you're gonna have to look at marker genes. So you're gonna perform a differential expression analysis between these clusters to see which genes are highly specific of each cluster to try to identify to which cell type they correspond to. So this bioinformatic pipeline, it involves a lot of different steps. So first, you filter the cells to remove a cell that were, for instance, broken. You filter genes to remove genes that didn't get enough read. Then you have to normalize your data. You do a dimensional reduction plot, then a clustering. You look at differential expression, and you do a functional enrichment of the differentially expressed genes. And at the end of the day, you obtain a cell type assignment for each of these clusters. So you see that it involves a lot of complicated steps with a lot of fine-tuned parameters to recover this information of which cell type your cells correspond to. So yeah, just to see how familiar you are with single cell data, we have a Google clap. So I should be able to take over from here. Yeah. Okay, so here, what is the information that can be obtained from single-cell RNA-seq data? So is it launch the question mark just when I click, or do we have to click somewhere to
It's
trigger?
launched, and if you click on the little histogram on the left, it hides answers.
Okay, so yes. And I don't see when does it stop.
Me neither. Maybe this one doesn't have a timer.
Okay. So yeah, what is the information that can be obtained from single-cell RNA-seq data? And then we can discuss the different options that are proposed. So people are still answering. I give you a bit more time. Okay. Let's go over the answers. People are still answering. Okay, let's have a look. So, differential gene expression by cell type, yes, that's something you can obtain for sure. Cell type marker genes, yes, indeed, that's what I just presented. So, other information that you can obtain, cell type composition of tissues, yes, something then you can obtain. So you take a sample from a tissue, you go through that tissue. So if you have a cluster that doesn't fall into a known cell type, and you look at the differentially expressed genes, and you see, oh, that this cell type, this immune cell type, for instance, in immunology, they discover new cell types all the time. So you see, oh, those genes express we never encountered before. Alternative splicing by cell, none of you answer that. You cannot obtain alternative splicing information from droplet-based technology. So because it's not full length, you only have access to the 3' end of the gene. There are some weird protocols allowing you to get droplet-based data full length, but it's very unusual. So you don't obtain alternative splicing information. Cell lineage, yes, you can obtain cell lineage information, basically by recreating a tree of the cells in your sample. And RNA velocity by cell, that's also an information you can obtain by looking at unspliced RNA. You do have access to some unspliced RNA, even though you're supposed to only select mRNAs with a poly A tail. You still retrieve unspliced RNA and can do RNA velocity, so to see to which states the cell is gonna differentiate next, or where it has been differentiated from. Okay, so apparently you are quite familiar with single cell data. Okay, and then once you get this analysis done, you can present the results. So there are different resources allowing you to interact with single cell experiment data. And so, yeah, here you're going to be able to see the different clusters and see to which cell type they've been allocated to. And then on top of that, you can look at gene expression. So here, for instance, it's the APOC1 gene. You can see that it is quite specifically expressing that cell type here. Okay, so then that's how you can visualize single cell data. So there are a lot of challenges regarding integration of single cell data. So the processing is going to depend on the protocol you use. So whether it was a single cell protocol or a single nuclei protocol where you're going to have unspliced RNAs, depending on the protocol, the barcode, the UMI positions are going to change. The length of the barcode of the UMI are going to change. Then the annotation is going to depend on your clustering. So how you process your data, the parameters used to do the clustering. So taking again the example of the fly cell atlas, because the fly cell atlas we did study very deeply. So they provide two types of clustering, a stringent clustering and a broad clustering, because yeah, they perform a first clustering and then they take some cluster and they reanalyze them very, very precisely. And we identify some cells that had a completely different annotations based on the clustering. So some cells were annotated as ovary cell in the stringent clustering and as neuron in the broad clustering. So you can see how even for the authors of the dataset, all of these steps are very complicated and challenging. And then it's very difficult to make single cell dataset analysis fully reproducible. Because when you go to Geo or SRA, you're going to find most of the time only the FASTQ file. But then really to make that fully reproducible, you need to find the information of barcode, the barcodes mapping to the clusters, the cluster mapping to the cell type. And so most of the time you don't have this information available in the primary repositories. You have to go to the paper or even contact the authors sometime to retrieve all this information. So I have a WooClap here, again, as a base for discussion afterwards. So the WooClap is like, yeah, here, what information do we need to recover from a published dataset to display the author cell type annotation on the clustering plot? So to be able to provide a clustering plot with cell type information, as I showed you, what are the information you need to retrieve to be able to display that to the users? Okay, so here I launched the WooClap. So yeah, what are the information that you need to be able to display this clustering plot with cell type information? And don't worry if you don't see exactly the answer. It's a bit like it's more a basis for discussion. Okay, so what did you answer? Just give me a nice cluster visualization. I always like this answer. It's like, yeah, it's complicated. So you pretty much gave all answer except library preparation protocol. And I think there are no wrong answer here. So let me explain. It depends where do you start from. So if you have access to the unprocessed raw data, then you're gonna need the library protocol information to do the read alignment to the transcriptome. And from there, you're gonna have a gene expression count matrix for each cell. So a lot of database, they start from here. They start from the gene expression matrix, gene expression count matrix. So if you start from here, you don't need the raw data, you don't need the library protocol information. Then if you start from the gene expression count matrix, you're gonna have to do the clustering yourself, and you're gonna end up with the coordinates of each cell in the dimension reduction plot that serves as a basis for the clustering. And you're gonna have maybe access to the cell type information for each cell. So for instance, that's how works the cell by gene database. They ask authors to deposit a data set with coordinates of each cell in the clustering plot and cell type information for each cell. So then they're good. They can display the cluster plots with the cell type information. If you do not have this information, as it is the case in Bgee, because we don't have authors depositing their data to us directly, we do go to their paper to find this information. So what we do have to do is that we have to find the association between cell barcodes to clusters and retrieve the information of cell type for each cluster. And then we are able to give the cell type information for each cell. So in Bgee, we start from here. We start from the association between barcodes to clusters and from cluster to cell type. And in Bgee, we do not provide a clustering plot because either the authors provide the coordinates of each cell in the clustering plot that they did produce themselves, or we have to redo a clustering analysis automatically. And a clustering analysis done without fine-tuned parameters is not going to look like what the authors provided in their paper. And we don't want to do that. We don't want to provide a clustering analysis that is not trustful to what the authors did in their analysis. So in Bgee, you're going to be able to retrieve cell type information for each cell, but we are not going to display that on a clustering plot. Okay? So I think it shows you the complexity of this data, and it depends of where do you start from, the information that you have access to. Okay. So in Bgee, we provide cell type information for each cell. We do not display the clustering plot. Yeah. Okay. So now that's how we retrieve data, how we treat data. And then in Bgee, on the gene page, as Mark showed you, we show conditions where any genes, the gene you are interested in, is active, okay? So it really look like in situ hybridization data. In situ hybridization data, you see directly with your eyes spots where a gene is expressed. And that's basically what we provide in Bgee on the gene page. We show you conditions where the gene you are interested in is expressed. So it's important to detect active signal of expression for filtering non-expressed genes, for instance, before performing a differential expression analysis. It's also interesting for studying condition-restricted genes, for instance, sex-biased genes that are expressed in one sex and not in the other. And also simply for answering the question, where is my gene expressed, okay? So a lot of studies, paper mention express genes, but they do not explain how to get there. So how do you do that? How do you determine that a gene is expressed over the stochastic transcriptional noise, over the technical noise of the protocol you are using? So I'm gonna focus on RNA-seq data. And so most of the time, authors to say, okay, I'm gonna look at the genes that are expressed in the condition I'm studying, they use an arbitrary cutoff. But an arbitrary cutoff, it's unlikely that it's going to fit all the conditions that are being studied. And there is little consensus of what is a good threshold to say, okay, look, these genes are expressed. So I have a book lab to ask you, what do you think is a good threshold to consider that the gene is actively expressed? Uh, yeah. So yeah, if you can answer the woo clap, yeah, what is a good threshold? Is it like as soon as you get one read map to a gene, 10 reads map to a gene, or 1 TPM? And it's interesting to see how little consensus there is on this value. Let's see the result. Okay, so I mean, why not? I also seen like answer one count, like as soon as you get one read, okay, maybe more than one read to be more confident. Relative to some baseline, yeah, that would be great. But then how do you identify that baseline expression level? So I see 1 TPM, 2 TPM. So 1 TPM, 2 TPM is the usual threshold used in a lot of paper. Okay, my gene is going to be considered expressed if I have more than 2 TPMs. 5 TPM, that can happen. It's more unusual, I think, because 5 TPM starts to be quite a high expression already, or two-fold change. So yeah, when you do a differential expression analysis, maybe people are gonna target only genes with a two-fold change of expression. But here, if it is you're comparing conditions, what if you don't compare a condition? Just look at, okay, give me the genes expressed in brain. You don't have anything to compare to. So we can see already from the answer that there is very little consensus about what should be used, okay? So I'm going to show you what we do in Bgee, okay? And here to show you, so usually the usual threshold is 2 TPM. So this is the red line here. And here, this is the density distribution of genes in 4 different libraries, in dog liver in blue, human blood, human brain, and in C. elegans embryo. And you can see how the distribution changes between these different libraries, even in human, like human brain and human blood. And here, if you consider 2 TPM for these 3 libraries, you're going to consider that almost no gene are expressed. And in this C. elegans library, almost all genes are going to be considered expressed. So those are not the typical distribution that I'm showing you, of course, because most of the time 2 TPM work. I'm just showing you that if you start integrating thousands of libraries, this 2 TPM threshold is not going to work. So you are not going to be able to say, okay, these are the genes expressed, these are the genes that are not expressed. So what we noticed is that when you look at the distribution here the distribution, the density distribution of genes in a typical RNA-seq library, what we see is that it's very typical to see a left shoulder here and then a right shoulder that is very high. And in this paper that starts being like a 15-year-old paper, they noticed that the genes that are on the left shoulder here, they correspond to non-expressed genes. They look at chromatin opening state. They checked every sign that the genes are expressed or not, and they see, okay, these left shoulders, they correspond to genes that are not actively expressed, okay? And you can still see reads from RNA-seq library mapped to these non-expressed genes. So what we noticed in Bgee is that this left shoulder of non-expressed genes, they match closely a distribution of intergenic regions. So intergenic regions, they are not supposed to be expressed. You should not find reads mapped to them in RNA-seq library, and still you find some reads mapping to intergenic regions. So we're like, okay, and what if we were using reads mapping to intergenic regions to estimate the baseline that some of you answer in the WOOC lab, to estimate a baseline of at which point gene starts to be actively expressed, okay? So this is what we do actually. So we use intergenic regions, so non-expressed genomic feature, and we compare, then we do a distribution of these non-expressed genomic features to obtain a p-value that your gene is actually expressed in that condition. Okay, so we did that. So we designed a method using intergenic regions, and then we evaluated how good our method was to estimate that the gene was expressed or not. So we used different species. We use different methods to estimate whether genes were expressed or not. In human, we use epigenetic markers. In mouse, we use ribosec data. In Drosophila, we use data from model organism databases to have the gold standard of saying, okay, those are the genes that are truly expressed. And we compare to our method of evaluating the distance from your gene expression to the expression of intergenic features. And we obtain very good results. We compare to other methods trying to identify genes actively expressed. And so we have a really good ROC curve, but the question is more, it's okay, it's nice to have a good ROC curve, but what is the p-value you're supposed to use? Usually we use a p-value of 0.05. And so ideally, when you use a p-value of 0.05, you should be at that point in the ROC curve, because that's really the best false positive, true positive ratio, right? So, and that's what you see in Bgee with a p-value of 0.05, the default p-value that you're going to use is going to be in that area almost all the time. And we show that because we compare to other tools that have very good ROC curve, but when you use the default threshold they recommend, you are not in the good position in the ROC curve. Like with that tool, you can see you are far away from the inflection point. Here as well, you're far away from the inflection point with that method as well. So with Bgee, you get a good ROC curve, and with the default value, you are at the inflection point most of the time, okay? So we think we have a very good method now to detect genes actively expressed in any sample. So I have a WOOC lab for you to think about this method of using intergenic regions to detect a baseline of expression level. So what could be the limitation of using intergenic regions as a reference non-expressed genomic feature? So yeah, can you think that could be a problem as like actively expressed? I see some of you are still answering, so I wait. Okay, let's go over your answers. So I go over the each point. So they will not be expressed. So yeah, intergenic regions are not supposed to be expressed, but that's the point actually, that even though some genomic features are not expressed, we do see them. We do have reads mapping to them, whether it is because the transcriptomic machinery of the cell is stochastic. And so, yeah, it does generate some RNA part for non-expressed genomic feature, or it comes from the technology that generate noise, okay? So that's exactly the point. Intergenic features, they are not expressed, so we want to use them to estimate this baseline of expression. Annotated genes considered as intergenic. Yes, so a lot of you provided that. It's very well annotated. It's very unlikely that we will have intergenic regions that contain unannotated genes, but in non-model organisms, we do see that a lot. So what we do is that we have a first step where we remove intergenic regions that are likely to contain unannotated genes. So we take all the libraries that we have, and we map reads to intergenic regions, and we remove intergenic regions that have a too high level of expression. And so we're like, okay, these intergenic regions, we suspect they contain unannotated genes. So we remove them from our analysis to estimate this baseline expression afterwards. Inconsistency of intergenic expression levels between experiments. Yes, it is inconsistent, but that's exactly the advantage of this method, is that then we're gonna have a threshold that is adaptive. So adaptive per library. So in one library using these intergenic regions, we're going to find that the threshold of expression is 2 TPM. In another library, it's going to be 0.2 TPM. In another library, it's going to be 10 TPM. And that's exactly the advantage of that method, that I showed you earlier, a different density distribution for different libraries. And you could see that the threshold would have been very different from each of these libraries. So that's exactly this advantage. Yes, the expression level is inconsistent between libraries, and that's why we want to use it to have an adaptive threshold. RNA-Seq reads cannot be mapped to them. Yes, that's what I would have expected, because from the library protocol, it's supposed to target only mRNA with a poly A tail, but that's not in reality what happened. In real life, we do get reads mapped to intergenic regions. So we do get reads mapped to unspliced mRNA, even though they would be all supposed to be spliced, you know? Expression level too low to be detected. It's not the case. We do have expression level high enough. It is a problem in droplet-based single-cell data when you look at each cell individually. So I will get back to that. It's not a problem in bulk RNA-seq. It's not a problem if you look at expression in a cell population. Okay, we do have enough reads to detect expression signal for intergenic regions. Okay, so yeah, you provided an interesting answer. The majority of your answers are totally correct, actually. But that's what we leverage, actually. Okay, so getting back to single cell data. So single cell data, you have like two types, two main types of protocol. You have full length, like smart-seq data that are full length. So you have a good sequencing depth for each cell. So each cell, you're gonna have hundreds of thousands of reads. And it's gonna look like very similar to bulk RNA-seq. So here it is the density distribution for a smart-seq library, which is full length with a good sequencing depth for one cell, okay? So one library is one cell for smart-seq data. And here you can see in red the distribution of protein coding genes, and in blue the intergenic region. So you see it's not great. It's not as good as for bulk RNA-seq data, but you can still use intergenic regions to distinguish between protein coding genes that are expressed or not. So still good. We can still use our method on smart-seq data. Um, So yeah, here it's on full length smart cell data. We compare bulk RNA-seq to single cell data, and we look at the percentage of protein coding genes that we detect as expressed. And it's lower than with bulk RNA-seq, but we still get some signal, okay? Now, if we look at droplet-based data, if we look at one cell, so here it's one B cell in two different libraries. So it's two different cells, sorry. And the signal is really not great. So here it's a feature receiving no reads at all. And here you see a little peak here, and we zoom on that peak, and that's what we obtain. Reads mapped to protein coding genes, to genic regions, intergenic regions, and here there's no signal. We cannot distinguish. You see the intergenic region, they completely overlap the protein coding genes. How can we say what is expressed or not? And it's because in droplet based, you get really not a lot of reads per cell. You get a lot of dropout, a lot of features that have just zero reads. So how can we do? So what we do in Bgee is that we don't look at the expression really per cell. We first do a pseudo bulk. We aggregate all the cells from a same cell type in a library. So that calls pseudo bulking the data. And here we retrieve some signal. So again, you get like genic feature, protein coding feature, and intergenic feature. The reference intergenic that we filter, the intergenic feature that we know that don't include unannotated genes. And here, yeah, we get some signal again. We are able to distinguish between protein coding genes expressed and protein coding genes not expressed. So it's what we do in Bgee. We look first at the cell population, as a cell type, and we say, okay, this gene is expressed in this cell type, and then we can go back to the individual cell. Okay, in this cell, I have one read mapped to this gene, so I know it is expressed because I know it is expressed in this cell population. But it does not really matter, but in Bgee, we're going to provide cell population information, okay, about the gene expression because it's highly reliable. So getting back to WooClap, question. So yeah, so here I have actually two questions. What is the main difference between bulk RNA-seq and smart-seq RNA-seq? It's a full-length protocol. Okay. So yeah, first question about the difference between bulk RNA-seq and smart-seq RNA-seq, so that we can discuss and see the point that might have been misunderstood. Okay, so just say what you think. No problem if you don't really know. So yeah. Well, and maybe I go a bit fast on this question because then I have the same question, but about droplet-based RNA-seq data so that you can have an idea of really the differences and the complexity for the analysis. So, okay, maybe I'm just going to stop this question now and we spend more time on the droplet-based, okay? So, difference between bulk RNA-seq data and smart-seq full-length data. So, no unique molecular identifier in bulk RNA-seq data. Yes, that's correct. And you do get UMI. So in smart-seq data, yes, you get the barcode. You don't get a barcode. So sorry, in smart-seq data, you also don't have a barcode. You don't need it, but you do get a UMI. No, okay. I apologize. In smart-seq full length, you don't have a UMI because it is full length, so it's not specifically 3' end with a UMI attached. So you don't get a UMI also in smart-seq data. High dropout in single cell data. So you do have a dropout, but it's less the case for smart-seq data, okay? Because one library is one cell, you can still get to hundreds of thousands of reads. So you do have dropouts, but it's not so much an issue, okay? Full isoform reconstruction, not possible. So again, with smart-seq data, it's full length. The entire transcript is gonna be sequenced, so you can do full isoform reconstruction. Reads not stranded, it is stranded. No, you can actually get the exact strand in smart-seq. Reads cannot be mapped using a pseudo-aligner. None of you answer that. You absolutely can map using a pseudo-aligner. Okay, so now I get to this question with droplet-based data. Okay, so I don't know if you want to answer, or maybe I can just go through again because it's the exact same question. So with droplet-based data such as 10x, so you do get both a cell barcode and a UMI. So it allows to really know the exact number of transcripts without bias from amplification. So we know that when we do the amplification, some transcripts are biased, they are going to be more amplified, so you're going to overestimate their expression. And with 10x, you get a cell barcode and a UMI so that you don't have like amplification biases. High dropout in single cell data. So then, yes, that's the case. You have a high dropout. It's very hard to estimate the expression for each cell. So usually people, as soon as they see one UMI for one gene in a cell, they say, oh, it's expressed. It can still be noisy. Full isoform reconstruction, yeah, it's not possible with 10x because it's only 3' N. Reads not stranded. It can be stranded as far as I see. And reads cannot be mapped using a pseudo-aligner. Yes, it can be mapped. This pseudo-aligner, they take into account the cell barcode and the UMI. So yeah, they adapted their aligner so that you can use it on 10x data as well. Okay? So now I'm going to show you. So I focus really on RNA-Seq because I think that the data type that most people are using nowadays, okay? But then we have like different experiments, different data types in Bgee. So how do we do to integrate all of that? So I focus only on bulk RNA-Seq and single-cell RNA-Seq, but Mark mentioned we also have like in situ hybridization data, Affinimatrix data, but that's nowadays we actively developing the work on single-cell RNA-Seq and still bulk RNA-Seq. So again, so we use a distribution of expression level of intergenic regions, and we compare expression of genes to this distribution of these non-expressed genomic features, and we obtain a p-value, okay? So for bulk RNA-seq data and single-cell full-length data, it's really the same method that we use, okay? For single-cell droplet-based data, we cannot have enough power per cell, so we pseudo-bulk all the data from a cell type in a library, and then we use the same approach, okay? And so at the end of the day, it means for each gene, each sample, we obtain a p-value of expression, okay? And then how do we manage to reconcile information between different experiments, different libraries, different data types like that? So I'm going to show you an example. It's from the latest Bgee paper. I show you here an example of single-cell data in 3 species coming from 3 different experiments. So human, mouse, fly. So we get an experiment in human tissue CA1 sample at 51 years old, and that we have one cell population here in the clustering pod that is oligopalin lama2. In mouse, we're gonna see that it's a tissue olfactory bulb. We have information of the age of the mouse, and the cell population here was Astro3. And in fly, we see that the tissue is brain. We have age of the fly, and that cell cluster is Enschitting-Glia. I never know how to pronounce that. Okay, so that's the information that the authors provide. So the first step in Bgee, so that's what Mark presented. We're gonna map to ontology terms. So this tissue CA1 it's mapped to the human CA1 field of hippocampus. That life stage we map to the human developmental stage ontology. That cell population, it corresponds to that term in the cell ontology oligodendrocyte. We do the same in mouse. So the tissue is olfactory bulb, the cell population is astrocyte. And same in fly. Okay, that was this term brain, and the cell population correspond to the cell ontology term and sheeting neuropal associated glial cell. So you see how we go from the author's free text annotation to a highly standardized ontology term. So that's the first step that our creators, annotators do. They spend a lot of time retrieving the free text information provided by the authors and map that to highly structured ontology, okay? And then we're gonna propagate this information into the ontology. So all of those terms, CA1 field of hippocampus, olfactory bulb, they all belong to the brain. So at some points, we're going to propagate in the ontology and we're going to reach this common term, brain. All these three experiments were performed in the brain. You could not compare at the hippocampus level or olfactory bulb level because, yeah, they were not aligned. But propagating into the ontology, at some point, you reach a common term, brain. And then we do the same for the cell type, for instance, oligodendrocyte, astrocyte, and sheeting neuropathy associated glial cell. They are all subtypes of glial cell. So again, it was not comparable taking just the information like that, but using the ontology at some point, you're going to be able to say, okay, these cells, they are all coming from the brain and they are all glial cells. And then you're going to be able to perform comparison between species and between experiments doing this propagation like that. So you still have access in Bgee to the very precise information, and you can still compare oligodendrocyte between any experiment or species providing this information. But you can also compare at another level so that everything is comparable when you propagate in the ontology graph. Okay, I hope it was clear. Another advantage, I present another way of looking at that. So here it's about like looking at the pancreas. So pancreas, you have like two term, endocrine and exocrine pancreas. And imagine, for instance, like we have three genes and we have three genes with different level of information. One gene, we had information in exocrine pancreas, another one in endocrine pancreas. And the third one in pancreas that can come from in situ hybridization data. They look at expression, very specific expression of different genes. So, and in the graph, it's going to look like that. Like one gene is going to have information in exocrine pancreas, another gene here, endocrine, and the third gene only in pancreas. So we have no comparable data here, but we're going to use graph propagation so that at some points, using the structure of the ontology, we have information for the three genes in the pancreas. So, and that's how leveraging ontology reasoning that Mark mentioned, we can get comparable information for any gene in any species. Okay, so getting to the WOOC lab to see what you think about that, how you understand it. So the graph of condition generated in Bgee. So several proposition asking you your understanding of how we use this graph of conditions, how we use these ontologies to make this comparison, to propagate expression information in the ontologies. And again, it's more a base for discussion, okay? Hide the results so that you're not influenced. Okay, let's look at your answers. So a lot of you answered, consider all permutation of an identity depth stage sex trend. Yes, that is a good answer. In this example I show you, I focus only on the anatomy, but here in this example, it was also considering the developmental stage ontology, where sexually immature is part of fully formed. And you can see here that we use all the permutation possible. And that's why we call that a graph of condition. It's not just considering one ontology, it's considering all the ontologies and all the permutation of all terms so that we get a huge graph of conditions where everything can be propagated and compared to. The graph of conditions varies between species. So yeah, I didn't discuss that much, but Mark mentioned that, in Huberon, you get one anatomical term that is valid in multiple species. So we get one brain term that is valid in all species having brains, basically. But sometimes the relation between the organs, they can change between species, and we take that into account. So that graph of condition actually can vary between species, whether it is an insect or a primate or a rodent. The ontology can differ, and we take that into account. So it's used in anatomical ontology, yes, and we focus a lot on anatomy. It uses developmental stage ontology, yes, as well. Sex ontology and strain ontology. Sex ontology, yes, it's a very basic control vocabulary. We have male, female, hermaphrodite, and we have one root term that is any sex. So, but it allows to perform comparisons. So you have a sample in male, a sample in female. It's going to be propagated at some point to any sex so that you can compare the data if you are not interested in the sex difference. Using a strain ontology, it's the same. So we use the UniProt control vocabulary for strain, and there is just one root term, any strain, which is wild type. So yeah, again, it's just so that you can compare expression even though your samples come from different mouse strains, for instance. We don't have a complex ontology. Okay? I just check the schedule, what time we suppose. Okay, I'm supposed to have 8 minutes left. Okay. So then we integrate all p-values. For now, I showed you that we have like one p-value per gene and per sample. Then I showed you that we propagate this information in the graph of condition, but then how do we integrate all this information together? So usually when you have like multiple p-values, usually you do a multiple testing correction, so like a FDR correction, so that you get one p-value. So you get 100 p-values, you do a FDR correction, and you say, okay, my corrected p-value is that. But that corresponds to a question if you want to ask, okay, from all my samples, is there any sample showing significant expression of the gene? Then you will do a multiple correction. Or in how many samples do I detect expression of that gene? Then you're going to perform a FDR correction. But it's not exactly the question we ask in Bgee. The question we ask in Bgee is, from all the samples available, can I say confidently that this gene is expressed? And it's a different question to ask, and it has to consider all the p-values at once. And so we use that paper that was very interesting, a statistical paper showing that you can actually integrate all p-values by taking the mean and multiply by 2. And look at the paper if you want more information about the statistical validity of that. But that's how we do. We don't do a FDR correction. We do an integration of all the p-values together to give you one answer to the question, is this gene expressed or not? So I'm gonna go a bit faster because I don't have much time to finish, okay? So again, just remember, so we get one p-value per gene and per sample. We propagate all those p-values, so thousands of p-values in the graph of conditions. And then for each gene in each condition, we average these p-values and multiply by 2 from this statistical paper. And we give you one p-value, that gene in that condition. Okay, but so p-value are good. It's good to know whether your gene is expressed or not, but we miss quantitative information, how high your gene is expressed, what is the level of expression of your gene. And for that, it's very difficult to compare TPM values directly from different experiments, even more complicated between different species and different protocols, bulk RNA-Seq, single-cell RNA-Seq. So we do use non-parametric statistics to estimate the expression level. So non-parametric statistics is based on gene ranks. So we rank the genes in a condition based on their expression level, and we normalize those ranks and we transform them to make them comparable. So for each data type, we compute weighted mean ranks based on the expression level, and then we normalize the ranks between the data types and between the conditions. And we also propagate this rank information as for the p-values. We propagate rank information in the graph of condition so that for each gene in each condition, we integrate all the information available. And at the end, these ranks, we normalize them into what we call an expression score so that it's easier to understand. So the expression score, they go from 0 to 100. 0 corresponds to a low expression, so no expression, to 100. This is the highest expression that we see in the species, okay? So I'm just gonna show you example using this expression calls and expression ranks. So if you go to the gene page of the APOC, one gene is in human. So this is a liver important gene used for digestive functions. So you can see that at the top, ranked by the expression score here, so you get the p-value, uh, and you get the expression score here. And so the highest expression is in the right lobe of liver, which is expected, okay? You can also see reported absence of expression. So the absence of expression in a lot of tissue where you don't expect the gene to be important. And just to show you, like now, if I go through several species in Bgee, you can see that for that ortholog genes of APOC1, in all the species, you get left lobe of liver, adipose tissue, liver, other species continue liver. That was the top terms for those orthologous genes in the different species. Okay, so I didn't select liver, I just took the top terms. Okay, and you can see that in almost all cases, for all those species, liver is at the top with a very high similar expression score. Even the expression score is very similar in all those species. Okay, so you can see how providing integrated calls of expression using a p-value and using non-parametric statistic using an expression score, propagating this information using a graph of condition, you can get like highly comparable expression information across species and across experiments. And then if I enter this list of orthologous genes in our tool called expression comparison, so it compares the expression of all those genes to tell you where the expression is conserved, you see that it gives you the answer that the expression is highly conserved in hematobiliary system and in liver. No surprise. Okay, so to summarize, okay, so to know the conditions where genes are expressed, first we curate data to anatomy, development, sex, strengths, and then we integrate all this information by generating a p-value of a signal of expression for each gene, each sample. We provide expression ranks also for each gene in each sample, and then we propagate in the graph of conditions all this information. And at the end, we integrate all those p-values, all these expression ranks to give you one answer per gene and condition. This is the p-value of expression. This is the expression level for that gene in that condition. And everything is comparable between species and between experiments and between everything. Okay? So that's it. So, stop sharing. If there are questions that were not answered, so I think the answer you asked, Mark answered them in written, right? Are there any questions that were not answered?
No, there were no questions in the doc, and someone raised the point about the human protein atlas. I use the human protein atlas. They compute the consensus expression across tissues, and so as I replied, I mean, human protein atlas obviously has proteomics, but also RNA-Seq. But the RNA-Seq, they do a quite interesting tissue specificity, but they don't do calls of is it actively expressed or is it just noise?
Yeah, but it seems
Now, just now a question, is it possible to study alternative splicing?
so for now in BG, we don't provide transcript level information. We are studying it in our group to see how we can provide this information in an integrated way. Because at BG we really focus on the data integration between multiple experiments and the comparison between species. So we have a PhD student working on that, but not yet. And about the comment on the human protein atlas, so I think it's interesting to see that these other resources also do this integration across experiment. That is interesting. And there is also a cell by gene, for instance, doing that for single cell experiments. But as far as I can see, it's still, I would say, naive in a way that just integrate log2 TPM. And so I think there is a lot to say about integrating just log2 TPM like that. It seems to work so far because they work a lot on human and mouse. on very specific experiments. So it seems to work just like that. I don't think it's going to work for very distant species. Um, so, I mean, but I see values in these different methods, in the different resources, and I think our strong advantage here is really the comparison between species across species. I think that's really the value you can find in BG.
So we have again a question
How
about
is
the expression of long non-coding RNAs and microRNAs.
So it's going to be exactly the same. So it depends on the protocols available for the library we create. So if we do annotate a library that have a protocol allowing to retrieve long non-coding RNA, then we're going to process them exactly in the same way. We're going to estimate the background noise using intelligent regions, produce a p-value, rank the genes based on the expression. And because we have a normalization step, it's going to take into account the fact that we're not studying all genes in the genome, all protein coding genes. It's going to take into account the fact that you were accessing only long non-coding RNA. So that's the normalization step in BG. How is the background noise handled in single-cell RNA-seq datasets when determining whether a gene is truly off in a given cell? So for a given cell, it's a bit complicated. So that's why in BG, we pseudo-bulk the data first. We aggregate the data from a cell population. So we pull all the reads from all the cells from a given cell type so that we have enough signal to say whether a gene is truly off or not. And then we can go back to the individual cell, but we don't produce a p-value per cell in BG. We produce a p-value per cell type, per cell population. What alternatives to intergenic regions do you recommend as a non-express baseline in expression threshold modeling? So we are submitting a paper describing our method. So we do compare to other methods. I think usually other methods, they use a Bayesian framework, or they use, I mean, I did see a variety of methods to do that. They all have their disadvantages and their advantages. I do think that our method, based on our benchmark, of course, I'm biased, but based on our benchmark and comparison, I think our method is really like working the best so far as we can see, because you can use it on one single library at a time. But the disadvantage is that you have to map your reads to intergenic regions. So you have to go back to using your FASTQ file and do the mapping to the intergenic region. So that's the main disadvantage of our method, I would say. So what alternative? The most widely used alternative is just to use an arbitrary cutoff of 2 TPM.
I will put in the document a link to some paper which has an alternative method, but in our hands, and we've also recommended them to collaborators, and they work really not so well sometimes. So because they're nice statistics, very advanced, but they're very complex, and then they fail to actually converge or work well.
How does Bgee handle conflicting expression evidence from multiple datasets within the same condition node? Yeah, so that's by the integration of all the p-values. So that's the statistical method I was referring to, where you really need to go check the paper why it is valid to do an averaging of the p-values, but that's this averaging of the p-values that take care of that. So if there is like one library saying like, oh, the p-value is super high, but then multiple libraries showing a super low p-value, then the one data point that was queued is gonna not matter so much, okay? So yeah, it really comes from the integration of all the evidence. And if it is half-half, then we're gonna have a high p-value. We're not gonna say, okay, it's expressed in that condition. And we do also put a weight on this p-value averaging. So library with a higher sequencing depth get more weight. So the better the library is, the more weight it's gonna have. So I hope it answers your questions. Okay, thank you. So don't hesitate if you want to write in the document. I think we have a break schedule now. I think we are okay. We are 5 minutes late. I think it's acceptable, and we're supposed to have a 10-minute break. So I propose we keep it 10 minutes and
Okay.
yeah. So 10-minute break. We come back around 11:05. Okay.